The next part is we're going to look at skeletal muscle contraction itself, including the process of excitation-contraction coupling. So now we'll think about skeletal muscle structure. So the skeletal muscle itself makes up around about 40% of the body mass of males and around about 32% in females. Contrasting that to smooth and cardiac muscle, which make up about 10% of the body mass in both males and females. We're then having a little look at crudely at a structure. You can see that smooth muscle is unstriated. So you can see in the skeletal muscle and the cardiac muscle that you can see up there, you can see patterns of dark and light bands. So if you're looking at it, you can see patterns of dark and light bands indicating that striation, but you don't see that in the smooth. As we've already said, the voluntary skeletal muscle is innervated by the somatic nervous system, whereas, of course, the heart and the smooth muscle is innervated by the autonomic nervous system. So, as I've also mentioned, even though skeletal muscle is categorized as voluntary, and that's because it can be consciously controlled, much of the skeletal muscle activity that's going on is subject to subconscious involuntary regulation. Like I said, a lot of that is involved in posture. So when you're sitting or standing, there are lots of muscles being activated, and balance and movement such as walking. These are all things that you don't consciously have to think about. So if we're looking at a single skeletal muscle cell or fibre, this is what you're looking at. So here is your individual skeletal muscle fibre. And the muscles lie parallel to each other in a whole muscle. And then if you're thinking about the nuclei, just so that you know, skeletal muscle contain multiple nuclei and an abundance of mitochondria. So as I said, the muscle fibers lie parallel to each other, as we can see in this image right here. They're bundled together by connective tissue. And the muscle fibers are grouped together whilst also being individually separated by this endomysium. And they're wrapped as a compartment into a fasciculi, which is protected by a connective tissue called perimysium. I hope I'm saying all these words correctly. And then you've got numerous of these fascicles. I'm terrible at pronouncing that, for which I apologize. And these are divided into compartments by internal projecting epimysium. Again, terrible pronunciation. You'll probably Google it and get a much better pronunciation than I'll give you. But that is basically a protective sheath for the whole muscle. So the muscle fibers or the muscle cells are made up of multiple myofibrils that extend the length of the fiber. So here you've got your muscle fiber or your muscle cell. And within that muscle cell, you've got individual myofibrils, and you can see all of these present at this point. And these myofibrils make up about 80% of the muscle fiber. They're cylindrical, and generally they're around about 1 millimeter in diameter. The myofibrils, if you're having a look at them, they display dark bands and light bands. So dark is A, so if we're taking a snapshot of this myofibril and then zooming in on it, you can see the A band. So that's looking quite dark by electron microscopy. And you can see the I band, which is looking quite light in color, hence why they're dark and light bands. And then these are actually composed of what are called thick and thin filaments. The thick filaments, they're composed of the protein myosin, and the thin ones are composed of the protein actin. And you've probably all heard of actin-myosin contraction because they are involved in contraction of the muscle itself. So now we'll have a look at the A band. And remember the A band, that's this bit, or if we're looking at the other image, this bit, and this is the dark band. And it consists of stacked thick filaments. And remember what the thick filaments are, or the thick protein? They're made up of myosin, but they have portions of the thin filament. So basically these A bands have got the protein myosin within them, and they've also got the protein actin. So actin, actually, if you're looking at this image, in this particular part is the blue bit, okay, to put simply, and the reddy brown bit is your myosin, okay? So you can see how the A band consists solely, all the myosin is in the A band, but you can also see how some of the thin actin is present within the A band, and some of it is also present within that I band. And where the thin filaments do not reach the A band, which is where that thick bit is, you're in that H zone. So you can see that here. Okay, so you can see that the thin filaments do not reach the A band in that H zone. And there are lots of supporting proteins holding those thick filaments together, and they're found within the M line, so that you can see that there. Okay, so there's the M line, but if we're looking within the A band, the M line is within the center of the H zone. So therefore, the M line extends vertically down the middle of the A band within the center of the H zone. So that's the A band as a kind of a summary. We're now going to look at the I band. So here it is. Here's that I band, and remembering from the I band, there's none of that myosin present within that, but we have some actin in there. The I band consists of the remaining portion of the thin filaments, which are made up of actin, that do not project into the A band. So there is the actin coming all the way through the A band and into the I band. And in the middle of each of these I bands, you can see this kind of Z-shaped thing, and that's a vertical Z line. And the Z line is basically a flat cytoskeletal disc that connects the thin filaments of two adjoining sarcomeres. So this is one sarcomere coming across, and then that's the next one going across. So it's basically a point where the thin filaments are joined together between adjacent sarcomeres. And the sarcomeres are the active components, the bits, the smallest component that can perform all the functions of that organ, in this case, skeletal muscle contraction. So if we're having a little look at that in a little bit more detail, here you've got the thick filaments, which is this red bit, okay? Here you've got the thin, and remember that's your myosin. You've got the thin filament, which is made up of your actin, and there's your Z line, okay? And your sarcomere goes from one Z line all the way to the next Z line, with the M line in the middle, remember. That's in the middle, that's joining the two myofibres together, the myosins. So you've got that joint in there. The area between the Z lines, like we've already said, that is called your sarcomere. And each sarcomere, therefore, as you can see from this, has a complete A band and half of each of the two I bands located either side. So it's got the complete A band, the dark bit, and half of each I band. Okay, and that, like I said, that sarcomere is the contractile unit, the smallest component that can perform all those functions. Just for interest, during growth, a muscle increases in length by adding new sarcomeres onto the end of the myofibrils, not by increasing the size of each sarcomere. So this image shown, whilst this is quite a complex concept because you've got thin filaments, thick filaments, M lines, Z lines, A bands, I bands, H zones, etc., etc. So it all gets a little bit confusing. This is actually quite a simplified image as it happens, because lots of other proteins also play a role. For example, there's a protein titin, and that is present along the length of the thick filament. Remember, that's the myosin, and it helps to stabilize the position of the thick filament in relation to the thin filament. And therefore, it acts like a spring and greatly helps a muscle's elasticity. In other words, it helps a muscle stretched by an external force passively recoil or spring back to its resting length when the stretching force is removed, much like stretching a spring. So we're going back onto the thick filament again. So we're going into a little bit more detail because we're going to be thinking about contraction. So each thick filament has got several hundred myosin molecules packed together. So here are our myosin molecules present. And this is what it looks like in a little bit more detail again. And what you can see is it has these cross bridges, as they're called. And these cross bridges are going to torque and bind to the actin molecule. So the myosin head forms cross bridges. Like I said, they can interact with the thin filaments, which is really the actin. So therefore, you have an actin binding site, so that the myosin and the actin can bind together. But also you have a myosin ATPase site, because muscle contraction requires energy. So the ATPase breaks down the ATP to ADP plus the phosphate. So those are two very important sites. And then you've got the thin filaments. And we've talked about how the thin filaments have actin on them, and that's a major component, but they also have tropomyosin and troponin. And if you're looking at this image, the blue circles, those are your actin molecules. You can see the gray part, that's your tropomyosin. You can see the actin binding sites, and you can also see a troponin complex. And you can see the troponin complex is made up of 3 proteins in itself. So the tropomyosin, these are thread-like proteins that lie end to end along the groove of the actin spiral, because you can see the actin is moving through in a kind of spiral shape. And in this position, it covers the actin sites that would usually bind the myosin cross bridges. So therefore, it's blocking muscle contraction. So in other words, you've got an actin binding site in the myosin that wants to bind the actin, but it can't bind the actin because there's tropomyosin in the way. It's blocking the myosin binding sites. So we have to get rid of that or move that tropomyosin before actin and myoglobin can bind together. Troponin, like I said, has got 3 polypeptide units to it, and so therefore it's binding to tropomyosin, it's combined to actin, and it can also bind to calcium. So when troponin, and as you can see at these troponin sites, you can see it's got calcium binding site. So when troponin is not bound to calcium, it stabilizes the tropomyosin molecule in its blocking position over actin's cross-bridge binding site. However, when there is an increase in calcium within the skeletal muscle, calcium binds to this calcium binding site, causing it to shift away, and the tropomyosin moves, allowing the actin to go on to bind onto the myosin. So therefore, in a relaxed state, actin cannot bind with myosin because tropomyosin is in the way of the cross-bridge binding site. But when calcium enters the cell and binds to the calcium binding site on the troponin complex, that tropomyosin moves and actin can now bind to myoglobin. So quite a complex little structure, if you like. So try and understand crudely what's going on with that. So a couple of revision questions for you, see what you can remember. Read them out, have a read and press pause, and then we'll go through them in a second. So in each of these cases, only one answer is correct. So the space between 2 Z lines constitutes the A, B, C, or D. The answer is D, the sarcomere, the contractile unit. Number 2, the function of tropomyosin in skeletal muscle include. Hopefully you've said it's B, acting as a relaxing protein at rest by covering the binding sites on actin, such that actin can no longer or can't bind to myosin. Okay, so now we're going to go into thinking about how contraction actually occurs in a little bit more detail. So when calcium binds to troponin, as we've talked about, it changes the shape such that tropomyosin slips away from its blocking position. But what we haven't really thought about at this point is how is calcium getting into the cells in the first place, into the skeletal muscle, to make this whole process happen. For now, I just want you to accept that this will happen, that on generation of an action potential in the skeletal muscle, that action potential travels throughout the muscle and down some structures called the T-tubules, but we'll come to that later. But in the end, it triggers calcium released into the cytoplasm of the muscle cell, and that calcium can go on to bind the troponin calcium binding site, which can then obviously help to move the tropomyosin away. So when that tropomyosin has moved, you then open up these myosin binding sites, and actin and myosin can bind at the cross-bridge sites. And that's what's shown here. Calcium comes in, binds to the troponin complex, and now we've got these nice little myosin binding sites that are nice and open up, so we can bind to that myosin cross-bridge. So the cross-bridge interaction between actin and myosin brings about muscle contraction by means of what is commonly thought of as the sliding filament mechanism. So again, here we come back to the images that we've already seen. So remember, a sarcomere, which is shown up in this part of the diagram, goes from one Z line to another Z line. You've got the myosin area, which it's got one end of myosin to the other end of myosin. That is your A band. You've got the H zone, where you don't have the actin or the thin filaments coming in. And of course, you've got the Z line, etc., etc. So during contraction, the thin filaments on each side of a sarcomere slide over the stationary thick filaments towards the A band center. So if we're having a look at this, this is the thin filaments. The thin filaments move in this direction, and they move in this direction, and they move in this direction, and in this direction. Hopefully you are following the little arrow, otherwise it'll get a bit confusing. And so as they move in all those directions, the A band, as you can see from this, stays the same width, okay, because that is the myosin, or the thick filament. But as you can also see, the I band becomes shorter. You can also see that the H zone becomes shorter. Remember, the H zone is just where the thin filament wasn't, and all of a sudden the thin filament slipped in or slid across, so that H zone has also become shorter. And because that H zone is shorter, and because the I band is shorter, the sarcomere becomes shorter. So like I said, the H zone becomes smaller as the thin filaments approach each other. The I band narrows as the thin filaments further overlap the thick filaments during their inward slide. But it's also important to note that neither the thin filaments change in length during fibre shortening. And neither the thin or thick, that should actually say. So now we'll go into this little bit detail. We've talked about how they start to cross over, but how does that actually happen? So here is our myosin crossbridge. Here is our actin myofilaments. We're only showing the actin. And in this one, we're looking at one dark blue. So it's basically been highlighted so you can try to follow it. And you can also see the binding sites there. So when actin and myosin make contact at a crossbridge, the bridge changes shape and it bends inwards. So you can see moving from this point, and here's your Z line, it basically pulls it in. And so you can see this, it's shortening, it's causing contraction. This is called the power stroke. And it acts a bit like as though it was on a hinge. This power stroke of a crossbridge pulls the thin filaments inwards, a small percentage of the total shortening distance. You then get detachment. And now what you can see, when it detaches at the end of the power stroke and returns to its conformational or its original conformation, so you can see how the myosin's changed its conformation back to its normal part, you get it binds to the next actin molecule, and the cycle repeats, pulling it in again, pulling it in again, pulling it in again. So basically you're getting repeated cycles of crossbridge binding and bending, and they go on to complete the shortening process. So the question really is, how does muscle excitation switch on this crossbridge cycling? So the term excitation-contraction coupling refers to the series of events linking muscle excitation, and therefore action potentials in muscle fibres, to muscle contraction, which is your crossbridge activity. So how does this happen? Well, skeletal muscles, as we know, are stimulated to contract by the release of acetylcholine at the neuromuscular junction. This is what we've already talked about. And at each junction of an A and an I band, the cell membranes dips to form a T-tubule. So here you've got your thick fibres and your thin fibres, and you can see, so they're your A band and your I band. And at each of these junctions, you can see how the cell membrane dips. So this green stuff is the sarcoplasmic reticulum. And in the sarcoplasmic reticulum, we have nice stores of calcium. Okay. And so what happens at each junction, you've got this A and the I band, and the action potential is coming along, and then it moves down these T-tubules. So you can see the action potential moving along nicely, then comes down these T-tubules. You then get local depolarization, and this local depolarization activates voltage-gated dihydropyridine receptors. And these receptors trigger the opening of calcium release channels, which are also known as ryanodine receptors. These ryanodine receptors are basically calcium channels. And so therefore, when you activate these calcium channels, you get calcium being released into the cytoplasm. And as we've talked about, we then get an increase in the calcium levels. That combines to the troponin complex. That therefore moves tropomyosin to the side, and that exposes the myosin binding site on the actin molecule, eventually going on to lead to skeletal muscle contraction, as we've just been talking about. So we've already said, so the myosin crossbridge has two sites. It's got the actin binding site that we've talked about, but it's also got an ATPase site. So in other words, it's involved in breaking down ATP to ADP and to phosphate. An ATP breakdown on the myosin crossbridge occurs before the bridge links with actin. So really what you've got in the resting stage you've got ATP. So here's our little structure. Here's actin, here's our troponin complex, and here's tropomyosin. At the same time, we've got myosin, the myosin cross bridge with ADP and phosphate bound. So it's ready to go in the ADP phosphate configuration. So in other words, the ATP has already been hydrolyzed and it's basically in a cocked position, just like the cocking of a gun, ready to go. The next stage that happens is the calcium is released because we've got the action potential going down the T tubules, opening up the calcium channels and leading to calcium being released from the sarcoplasmic reticulum. This binds the troponin, moves the tropomyosin to the side, opens up the myosin binding site on the actin, and now the activated myosin can bind to the actin itself. And then the power stroke occurs and the sarcomere contracts and ADP and phosphate dissociate from the myosin. So during the power stroke, this is actually during the power stroke itself, that's when the phosphate is released. And after the power stroke is complete, the ADP is released. And when actin and myosin detach from each other, the ATP is split by ATPase activity, and therefore that's energizing the myosin cross bridge once again. And then thus, on binding another actin molecule, the energized cross bridge again bends, and so on. And that leads to a successful contraction. So really what it means is actin and myosin remain attached at the cross bridge until another ATP binds to the myosin ATPase site. Okay, so remember, you get the actin and myosin have been joined at this point. We've done the movement. ADP and phosphate have been released, but they stay bound until ATP binds. And when the ATP binds, that causes detachment of myosin from actin. The ATP then gets hydrolyzed to ADP and phosphate by the ATPase activity. That then cocks it back into its, gets it ready into the ready position. So it goes on from not ready into the ready position. And then we're ready to go through the whole cycle again. So an interesting situation is rigor mortis. So why do we get stiffening of muscles a couple of hours after death? And this is because of the locking in place of skeletal muscles. So following death, the intracellular fluid calcium levels rise. That increase in calcium levels leads actin and myosin to interact because remember that we've opened up it's bound to troponin and it's opened up the myosin binding site on the actin. So the two of them can then bind. And at this point, myosin is already charged with ATP. Remember, it's already in this active position, ready to go. But the dead cells can't produce any more ATP. So actin and myosin, after the contraction, can no longer detach because there's no more ATP to come in and take the place of the ATP that's already been used, so to speak, or the ADP, phosphate. So the thick and thin filaments basically stay linked by immobilized cross bridges, leaving dead muscles stiff. And that's the process of rigor mortis. That's why it happens. But of course, the muscles don't stay stiff forever. So over a couple of days, rigor mortis subsides. Why do you think it subsides? It's basically because the proteins involved in the rigor complex begin to degrade. As the cells are dying, those proteins degrade, and therefore rigor mortis basically starts to subside. So rigor mortis usually starts a few hours after death, is usually complete by 12 hours, and then disappears after a couple of days. So what about relaxation of the muscle itself? So we've gone through this cycle and talking about how it contracts and contracts and contracts through that contraction process and binding of calcium and ATP. But when does relaxation occur? Well, it finally occurs when calcium is returned to the sacs of the sarcoplasmic reticulum when the local activity has stopped. Because removing calcium from the cytoplasm lets the troponin-tropomyosin complex slip back into its blocking position, and therefore this leads to muscle fiber relaxation. But it's important to note that the contractile activity outlasts the electrical activity that initiated it in the first place. So if we're looking at this image on the right-hand side, the bottom part is looking at membrane potential, so the voltage change against time, and whereas the upper bit is looking at tension, so contractile strength. or contractile response. So you can see, if you're looking at the stimulation, a single action potential in a skeletal muscle cell lasts around about 1 to 2 milliseconds. But the onset of contraction lags this. In fact, as can be seen in this image, electrical activity is complete before contraction even begins. And this delay between stimulation and the onset of contraction is called the latent period, which is shown up there. The time to peak, okay, that varies between muscle fibers, but averages around about 50 milliseconds. Okay, so here you've got the contractile strength going on, the tension, and it tends to peak at about 50 milliseconds. And this is a muscle twitch. Okay, and we're not going to go any further than this at this point. You can look at that later on yourself. So like I said, the muscle fibers peak at about 50 milliseconds, and the contractile response does not end until the sacs themselves have taken up all the calcium released in response to the action potential. And obviously this is a bit time consuming, so it takes round about another 50 milliseconds for this to happen. And even after the calcium has been removed, time is still taken for all the fibers to return to their resting state. So that's basically what we're talking about here. We're talking about calcium's getting taken, disappearing back into the sarcoplasmic reticulum, but the muscle has to go back into its resting state. So therefore, the entire contractile response lasts around about 100 milliseconds, which is much, much longer than the action potential duration of 1 to 2 milliseconds. And this is really quite important because it helps in the ability of the body to produce muscle contractions of variable strength, because you can add twitches to increase muscle contraction strength and therefore tension. Okay, so just a couple of questions to end. So have a read of the questions. Press pause first, obviously, because I'm going to do the answers in a second. Press pause, have a read, and then we'll go through them. In each case, there's only one answer correct, just so you know. What is the role of acetylcholine in calcium release? The answer, hopefully you got, is it stimulates cellular contraction by opening voltage-gated ion channels. So in other words, through the generation of action potentials that move through the T tubules and therefore go on to lead calcium release from the sarcoplasmic reticulum. So it stimulates contraction because it's opened voltage-gated ion channels, sodium ones initially and then calcium ones afterwards. Which of the following correctly identifies muscle components in order from largest to smallest? The answer is D. Fasciculus, muscle fiber or muscle cell, and then myofibril. Next question. Press pause. Okay, which of the following would tend to reduce the concentration of lactic acid that accumulates in a skeletal muscle as a result of contractile activity? Again, only one is correct. Increasing the concentration of glycolytic enzymes? No, that's not correct, because glycolytic enzymes are anaerobic because they're in the cytoplasm. Decreasing the oxygen supply to the cell? No, that won't either. Increasing the diameter of the cell? Nope, that won't make a difference. Increasing the number of mitochondria in the cell? Yes, that will obviously tend to reduce the concentration of lactic acid because the mitochondria are aerobic, whereas anaerobic respiration takes place in the cytoplasm and involves the glycolytic enzymes.